In order to produce haploid gametes, which have half the amount of chromosomes of diploid, individuals must have cells that undergo cell division that does not produce daughter cells that are genetically identical to the parent. Otherwise, the gametes would be diploid and a zygote formed from diploid sperm and cell would be tetraploid. Meiosis is where haploid gametes are produced. The two-stage process of meiosis reduces the number of chromosomes in the cell by half. One chromosome from each homologous pair, which are chromosomes that have the same genes but not the same alleles, is present in each gamete. Interphase is the longest stage in the eukaryotic cell cycle. During interphase, the cell acquires nutrients, creates and uses proteins and other molecules, and starts the process of cell division by replicating the DNA. Prophase 1. Chromosomes become visible, crossing over occurs, the nucleus disappears, and meiotic spindle forms. The nuclear envelope disappears. The synapse, also called synthesis, is the pairing of two homologous chromosomes that occur during meiosis. It allows matching up of homologous pairs prior to their segregation and possible chromosomal crossover between them. A crossover is an exchange of parts of the chromosome. Metaphase 1. It highlights the pairing of and the exchange of DNA between homologous chromosomes through homologous recombination and crossover at the chiasmata, which is a point at which paired chromosomes remain in contact during the first metaphase of meiosis, and at which crossing over and exchange of genetic material occur between the strands, also between the non-sister chromatids. Random orientation occurs here, and it is orientation of parental and maternal chromosomes on either side of the equator is random and independent of other homologous pairs, help with diversity with daughter cells. Anaphase 1. Anaphase 1 begins with the two chromosomes of each bivalent, which is the homologous chromosomes pair, and form synapses and separate and start moving towards opposite poles of cells as a result of action of the spindle. Notice that in anaphase 1, the sister chromatids remain attached to their centromeres and move together towards the pole. Telophase 1. Telophase 1 is the phase that the chromosomes have finished moving to the opposite ends of the cell. This will then be followed by cytokinesis, producing two daughter cells. Two chromosomes of each bivalent separate and move to the opposite poles. Prophase 2. The chromosomes are condensed. The centrosomes replicate and move towards the opposite poles. Spindle fibers grow outward from the centrosomes. Metaphase 2. It highlights the condensation of the chromosomes and the movement of centrosomes to polar regions of the cell. The cell is in metaphase 2 when the chromosomes align themselves along the metaphase plate through the pistillation of the spindle fibers. The spindle fibers are now attached to the two kinetochores contained in the centromere of the chromosome. Similar to the mitotic metaphase, the two kinetochores are bound to the spindle fibers, roam opposite poles, and they lie on the equilateral plane, readying for the chromosome movement towards the opposite pole in anaphase 2. Anaphase 2 are where the sister chromatids separate, and the centromere splits into two, which result into two separate chromatids. Anaphase 2 occurs in haploid cells. Now the sister chromatids separate, each chromatid reaches each pole, and at the end of the phase, each daughter cell has one sister chromatid. Telophase 2. A nuclear envelope forms around each set of chromosomes, and cytokinesis occurs, producing four daughter cells, which with each with a haploid set of chromosomes.